This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 303, recorded on September 19th, 2014. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today, right here on the Hudson River, Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
I'm chipper.
We are on the Hudson River, you know.
Yes, we are.
About, I would say, half a mile from it.
Well,
Less?
Yeah, less than half a mile, and we're actually almost in the estuary of the Hudson River.
What is an estuary?
It's the meeting of the fresh water from the river and the salt water from the ocean.
Oh, yeah? Where is it roughly?
It's right out in front of us. There is a tongue of salt water that actually goes down the Hudson. Actually, it goes up the Hudson, up and all the way to Albany, which is 163 miles from here.
Wow.
And you can find salt water right up to the weir, which is why they installed it.
The weir?
The weir.
Last week, before we recorded, remember there was a white thing?
A white slick.
So you told me to email the River Watch,
the river keeper,
and I did, and he answered, or she answered.
No, it's a he.
And they said, it's probably this.
Cool. Right.
Today there's more weirdness. There's a boat,
and
it's spraying this huge plume of water.
That's right.
Right?
That's right. Looks like a fireboat practice, but it's not a fireboat. At least it doesn't look like a fireboat.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
Can you see a river from your office window?
No, not from my office, but it's not very far away.
Is that the Huron River?
Mm-hmm.
Yep.
Yep.
The Huron River.
Has it started to snow there yet?
No. No.
You know, in Bozeman, they got snow already.
Did they?
Oh, yeah.
Oh, yeah, you remember Mark Young at the ASV
told us,
meeting.
yeah.
Isn't that crazy?
Mm-hmm.
Here, it is really a nice day today, right, Dickson? It's a
gorgeous.
little warmer than it's been all week.
It's a perfect day, to be honest.
No, it's not perfect because...
I would say it's perfect.
Well...
There's no humidity to speak of, and there are a few fluffy white clouds.
It's chillier than it's been. It's only 18 Celsius.
Well, you know what? I prefer that.
49% humidity. Maybe the humidity makes it feel a little warmer.
It could.
Let's ask our weather expert from Western Massachusetts, Alan Dove.
Good to be here.
Hey, Alan.
I guess the humidity affects the, the heat index, they're calling it now.
Yeah.
Right. Right.
So it's up here, it's a nice 62 Fahrenheit, 17 C, clear blue sky.
Nice.
Nice, just starting to get to be autumn here. And I'm about a mile and a half from the Connecticut River.
Right. Do you have a little hint of leaf turning yet?
Not quite yet. It's
okay.
It's going to be getting to that point pretty soon.
Yep.
I have leaves on the ground here.
Yes.
Not a lot, but
we've had a droughty fall, so
is that right?
Sometimes the leaves just turn brown.
Oh yeah, we've had some a few dry leaves falling, but it's not really. They're not turning and falling. They're just sort of falling.
Well, Dickson, September's half over.
It is.
Or maybe we should say it's half begun.
On the weekend, we're going to turn into fall.
That's the 21st?
Yeah.
Sunday. That's when this episode will be released.
That's right.
Yeah.
So I wanted to make two comments.
Yeah.
Our temperature is the same, and my app says 66 degrees, feels like 66 degrees. So I don't think the humidity's doing anything here. And the fact that everybody lives close to a river just reminds me of a key fact that I remember from elementary school is that civilization grows up around rivers.
Indeed.
Or bodies of water.
Indeed.
Well, that, in fact, the town of Springfield, Massachusetts, which I live right outside, the reason it exists is because it's right at the fall line of the Connecticut River, and that's where you build your mills.
Hey.
Connecticut River.
Yep.
Well, Sunday it's going up to 26.
Right.
Dickson.
Yes. But you should also mention the fact that the lower the temperature, regardless of what the percentage of humidity is, the actual amount of humidity in the air, the moisture in the air, is much less when it's cooler.
Yeah, of course.
Yes.
So it still feels comfortable, even at 47%.
I would like to mention that this episode of TWiV is brought to you by the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. Composed of over 20 virology labs, all centralized in one building in the heart of this great New York City. This department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to their program by the December 1st deadline. Interested postdocs are also encouraged to contact faculty of interest. For more information, please visit mssm.edu/MIC. I gotta tell you, I can vouch for this because that's where I got my PhD.
Yeah.
So our first academic sponsor.
Cool.
If anyone else is interested in sponsoring TWiV, we'd love to hear from you. You can send an email to twiv@twiv.tv. It helps us to do the things that we do, right, Dickson?
You betcha.
Awesome.
All right. Follow-ups. Kathy seems to have one.
Several, because I wasn't here for the picks of the week. And I really liked Dickson's second pick, which didn't end up having a link put in the show notes, but maybe we can put it in this week. It's a science article about self-programming robots, and it had really cool videos as supplemental material.
Yep.
I sent the ImmuneQuest game link to somebody before TWiV, a colleague who teaches immunology to undergraduates, and he said it was pretty addictive. He played for 45 minutes.
I also have a follow-up about ImmuneQuest, by the way. After the episode, they emailed me. Apparently, one of their developers is a TWiV listener.
Wow, cool.
So shout out to them. Thank you.
Because you had problems with your Mac version.
I had problems with the Mac version, and this pretty much defines excellent customer support. It's a free game, and they emailed me and said, "So what was the problem exactly?" So I described the problem to them, and they have issued an update. I have not had a chance to test the update, but if you're on a Mac and you were scared away from the game by my mention of a glitch, you should still try it out, because these folks are very on top of fixing stuff, and they may have fixed the current version.
Nice. That's great. I like that. I like when we get response.
Yeah. And I did watch the little bit of Wildcrats. They do seem addictive. Those are the things that start out with two real guys. The one I saw, they're on bikes, and then it goes into animated stuff. And that was really good. And then also follow up from our TWiV 300, where we had Mahul's pick about the NOVA program, Vaccines Calling the Shots. I watched it, and it is excellent. And I'm also providing a link where you can watch it live from the NOVA site. It's really worth watching.
Good. Yeah, I heard it was good. All right, we have a lot of follow-up today on Ebola virus.
You'd think it was a hot topic or something.
Hey.
This is. I think this is the most engaged any single topic has been, except perhaps XMRV.
Exactly.
Yeah.
I was just going to say.
So this, of course, is continuing in a big way, and we have a whole bunch of email. And these are largely dealing with transmission. So
Sure.
Let me take the first one here from Ted. First, wanted to thank you for several months ago introducing me to your TWiV broadcast. They are very informative and a good listen, especially now with the Ebola situation. Years ago, before finding your work, hearing there was a large Ebola outbreak, and then seeing an interview with a doctor like I saw on Fox News today would have made me call in sick and start stockpiling food, water, guns, hazmat suits, and prepping for the end. But after listening to your class on podcast and hearing your discussions on TWiV, I feel I've gained some knowledge on the subject, and I no longer panic. So I thank you and everyone on TWiV for that. It's a beautiful thing. This is exactly what we're trying to do, right? To counter all the negativity out there. What made me send this to you was, as I said above, this morning I listened to an interview with a Dr. Jorge Rodriguez on Fox News. He said that the virus is in all these people and it's constantly mutating. He said the virus is like a string of red beads inside these people, and all it takes is for one red bead to turn blue, and poof, it becomes airborne.
Oh, my God.
Like I said 5 years ago, the doctor saying this would have put me in panic mode. After TWiV, it's rather comical. If it is that simple of a process, how come in other Ebola outbreaks it never happened?
That's right.
If it's that simple, why don't the dengue fever virus, which the virus is in far more people than this current Ebola, say, hey guys, instead of having to wait for and travel with a vector, why don't we just make one of these red beads turn blue and we can go airborne and spread faster? I think you all need to get Dr. Jorge Rodriguez on your TWiV broadcast and have him explain to you all just how simple it is for a virus to mutate. And turn that one red bead to blue, and let's have it live on Fox News.
Oh, my gosh.
Keep up the good work. You all are the best at what you do.
Wow.
Well, this is an interesting topic, which we talked about last time, of course.
Yes.
Beads, huh?
You got an email from the Washington Post about 4 or 5 days ago, and as I was in Detroit, I had some free time, so I answered, and I had a nice telephone call with the reporter, and I just repeated what we had said on our Ebola broadcast, or podcast, I should say, about the unlikely events of this virus turning into something that it's not. It's doing very well as it is, so why should it want to switch anyway? But it doesn't have a mind of its own, obviously. So
I'm so glad you added that, Dickson.
Well, yeah, and then I recommended that she get in touch with Vincent, and apparently she did.
Well, she emailed me in the morning while I was in Georgia on Monday.
Right.
But
she got with Rich. I mentioned Rich as well.
And Rich is quoted in the article, and I'll put a link to it.
That's
coming up later. Kathy, can you take the next one, please?
Sure. Dell writes, Dear TWiV team, one of the letters from your recent episode, The Sky is Falling, seemed to take you to task for not being clinicians and therefore not really understanding the nefarious intentions of Ebola. As a clinician myself, I simply want to counter that impression by commending you for how clinically thoughtful and relevant your discussions are. TWiV is, after all, about viruses, the kind that make you sick. You seem to be meticulous about not making clinical statements about which you are unsure, but even that doesn't seem to happen often because you seem to do your homework before the show. We fight a constant battle against misinformation in this media-driven world, and you are fighting the good fight.
Wow.
As an example of this balancing wire, we walk with the media, and please forgive me, Kathy, for using your institution as an example. Several years ago, we were having an apparent increase in invasive streptococcal infections in our state. I was personally caring for 2 severe cases in one of our hospitals in Saginaw, Michigan, at the time. When I returned home that evening, I walked in to see a press conference on the news from the University of Michigan hospitals. The head of the ID department and the head of the hospital system were answering questions from the media about the one case of flesh-eating infection they were currently treating in their ICU. That was the day I realized that it is all about marketing, and I am not a marketer. I commend you for the invaluable contribution you are making to science and to medicine, and I am evangelizing your podcasts among my colleagues. Dell. And he's an associate professor of medicine at Michigan State.
Right.
So this is the Michigan State versus you, Michigan. Yeah, well.
Right. Fine with me.
Yeah, exactly.
Dickson.
Yes.
Can you read the next one?
I certainly can. Robin writes, how likely is Ebola to transmit by aerosol? Slightly more likely than for pigs to fly, and like bats, pigs are mammals. The walrus and the carpenter, and then they have of course, a link to that, and then she writes. Well, you have another Robin below that.
Same Robin.
Okay.
I don't know why that
come,
matters.
The walrus said, to talk of many things, of shoes and ships and sealing wax, of cabbages and kings, and why the sea is boiling hot, and whether pigs have wings.
Lovely.
I actually, right after we recorded that episode, I thought of exactly the same analogy. Because, we can't exclude the possibility that pigs could fly if you gave them enough evolutionary time or enough genetic engineering.
That's right.
But the issue is that what you got out of the end of either of those processes would not really be a pig anymore.
No.
Would
probably be a bat.
Should we worry about it?
I'm not.
There are those out there that will worry about that, though. That's for sure. And let them, because that's where they deserve to be, apparently.
Robin also sent a link to an article in the Extinction Protocol. U.S. government to buy 160,000 Ebola hazmat suits to prepare for full-blown global pandemic I should just say they're not Ebola hazmat suits.
No.
They're just hazmat suits, right?
Right.
Yep. And the U.S. government is constantly making large purchases of protective equipment for a variety of purposes. So, yeah.
Full-blown they're not preparing for a full... They probably need these all in Western Africa.
These look actually like disposable suits, so it's the kind of thing that you would order in a huge quantity if, you're the U.S. government. Yeah.
It's true.
All right, Alan, can you take the next one?
Stephen writes, kudos to Kathy Spindler for nailing the nature of a listener email, and by extension several other recent emails about the Ebola epidemic. TWiV should turn its troll detector up to 11. If there are important points you all would like to make, they can be made without repeating a list of what if and how do you know questions that repeat incorrect or misleading ideas. Mm-hmm. As Vincent said repeatedly, we and policymakers have to rely on evidence. What's the alternative?
What did you say, Kathy?
Oh, this was just one of those series of emails, and I'm sure I said we weren't... I think it might have been the one where I said we weren't trying to solve the whole problem in Africa. We were just trying to inform about Ebola virus and so on.
Did you also say, this is like the question, when did you stop beating your wife?
Oh, I did say that too.
Yes, you did. Right. You did.
I was playing that.
Very good.
I think that's what this is a reference to.
Probably.
Which is exactly the
Sure.
That's exactly what the troll was doing.
Now, I think a lot of these came in last night, which is really cool because they know if they get it in before recording on Friday, they'll get them read. Next one is from Mary, writes, greetings, TWiV team. Weather today is in the upper 80s and sunny in Colorado. Wow. That's nice. It was just this week.
Yep.
Thank you for sharing your expertise so that many like myself can better understand the world of virology. I became a TWiV listener after taking Virology 1 and 2 classes online.
Nice.
The CDC and WHO websites for Ebola statistics have been silent the month of September. Today's the 17th, about the number of cases and deaths in Western Africa. The approach is now focused on how to recognize the symptoms, prepare, and protect. Leads me to believe that the probability that it will, quote, jump the continent, unquote, of Africa is increasing to the point that this event is now probable. These agencies do not respond to inquiries. Would you recommend viable sources? Thank you. So I always go to ProMedMail, and they collaborate with HealthMap, and they have a very nice health map at healthmap.org/Ebola, and it's updated as of September 18th. And you can sign up for a daily email or a daily RSS feed. So you can look at the numbers. 5,335 people in West Africa have been infected, 2,622 deaths. And they have other news down there as well. They talk about, the restriction of travel and so forth, and where the transmission is the highest. So I would recommend that. Anybody have anything that they use? I think this is good.
That's great.
Yeah, that's a great source. And I would just add, I'm not sure which part. I guess it's the Ebola page on the CDC or WHO websites, but the CDC and WHO have been continuously updating data on this. I mean, I see even in my Twitter feed, I get tweets from the WHO saying, here are our updated numbers. So it's not that they're not putting this information out. It's, I guess, maybe an issue of which particular parts of their site they're updating or not.
So this page we mentioned last week, it's a new Ebola outbreak page at the CDC. There's a link in TWiV 302. They at the right-hand part of the page, they have, updated September 18th. 5,347 cases, 2,600 deaths. So that's up to date too.
Yep.
I mean, sometimes the numbers are not coming out quickly, so you have to be a little bit patient. But those are the best places, I think, to look.
Right.
This map that you sent, Vincent, also breaks it down by countries. And if you click on a dot on any map, then it gives a sort of a timeline of different news reports for that particular region.
Yep, it's very nice. Very, very nice. All right. That brings us to Dallas.
Yes.
And I think, whose turn is it? Alan?
Kathy? Who just read that? You just read that?
I just read it, so it's Kathy.
So it goes to Kathy.
All right. Dallas writes, TWivers, you can view aerosols from a physical chemistry viewpoint. A small liquid particle with containing virus, soluble organic material, and salts will rapidly evaporate in any air at less than 100% relative humidity. This will result in rapid osmotic pressure change, which could be very rough on virus particles not designed for desiccation. At 100% relative humidity, the particle with the salts will grow in size and fall out of the air column. In either case, epidemiological evidence would indicate non-aerosol transmission. Love the podcast, Dallas. P.S. We play these osmotic pressure games for biosecurity in some fish shrimp hatcheries by using saturated salt brine to kill most aquatic pathogens of all types on surfaces and equipment. Very effective against biofilms that can effectively be desiccated with brine in seconds, while being easy on the staff's hands, especially relevant in some developing country situations.
Now, so it's correct that the smallest droplets will produced by an aerosol, sneezing or coughing, they'll dry out, but they have to be the right size so that they can go distances. And it does depend on the humidity, and Palese and others have shown that humidity is important for transmission of influenza, because when the humidity is too high, the droplets get too big and they fall to the ground close to you.
Yeah.
And if it's too low, they dry out. So winter seems to provide the right combination of low humidity and coldness to allow the drops to go further. And we have more coming on aerosol or airborne transmission.
Yep.
All right, are you next, Alan, now? Yes.
Yes, now I'm next. Yegor writes, Dear TWiVers, you discuss at length the very unlikely possibilities of Ebola becoming transmissible by airborne route or by mosquitoes, but have you talked about the more real possibility that it will become attenuated in the course of the current epidemic? Perhaps I missed it. If the virus becomes less pathogenic while preserving the ability to transmit between humans, that may lead to serious problems. In the worst-case scenario, the virus may even become endemic. I think attenuation of the myxoma virus in Australian rabbits is one example of this happening. The virus was extremely pathogenic at first, but now it's relatively mild, although I'm not sure how much of the change in pathogenicity is due to virus attenuation and how much is due to selection on the host. Probably both happened. At the moment, people become infectious when they already have very serious symptoms, and one can hope that those symptoms are required for transmission and a less pathogenic virus will simply become non-transmissible. But maybe not. I would imagine that at early stages of the disease or during a mild disease, sexual contact may provide the necessary contact with bodily fluids and enable transmission. Are there known cases of Ebola being sexually transmitted? Thanks for your excellent coverage of the Ebola epidemic, Yegor, who is a senior science officer at the Global HIV Vaccine Enterprise.
All right, so the Australian rabbit story is the classic one where they introduced a virulent virus to kill the rabbits, and within a couple of years, both the virus and the rabbits had mutated so that the virulence was less. I think initially it was 99% lethal in the rabbits, but then after those 1% of the rabbits that lived after that first year, then more resistant. However, it didn't go to zero. It's still about 50% lethal. So it hasn't gone away. And Frank Fenner studied this extensively in both the rabbits and the virus. You would think that this would happen in most hosts, that viruses would evolve to be minimally virulent and maximally transmissible to preserve the host long enough to transmit, but we don't really see that happening. And, I've heard that the virulence of HIV is becoming attenuated slowly as it's passing through people, but I'm not sure that everyone subscribes to that. You would know that. You work at the HIV enterprise there. So I'm not sure, again, we can't predict what's going to happen with Ebola, but I don't know what the selection would be for it to be less virulent, because it's transmitting well by contact because of the nature of the illness, the seriousness. You contact other people, healthcare workers, family members, and so forth. So,
is that how we presume the bats keep it going in their populations?
How
by
is that?
By direct contact?
I believe so, but there could also be an aerosol component in caves,
or perhaps
airborne.
Sexual transmission. Bats are very promiscuous.
That I would not know. I don't know that at all.
I watched a documentary once on bats showing, it was a rather graphic scene of a male bat taking advantage of a series of asleep female bats hanging upside down.
It could be.
And that male bat from one female to the next and inseminated all of them in a very short period of time.
I don't know.
So I know that there is aerosolization of rabies virus in some caves.
Well, that's a controversial topic. I just did a TWiV in Georgia with,
okay.
with a fellow. Did you know Zhen Fu, Kathy?
Yes, very well. He was on one of my students' committees, and we had some joint lab meetings together.
So I had him, and we talked extensively about rabies, and I said to him, how is it possible that rabies would transmit by aerosol? And he laughed. He said, there's some anecdotal evidence of aerosol transmission in caves. He said, but it's not really believed, and
good.
It's not very good. Okay.
Excellent.
And in that case, I mean, you're talking about aerosol transmission from bats to humans.
Yes.
Yeah. But now, whether it goes...
So the question is how a virus gets between bats. If it's endemic to bats, they do have, even though they're mammals, they have quite different biology from humans. And I think, didn't somebody send us a paper on this? About the temperature changes in bats as they're flying versus resting, and
so that was a paper we did on TWiV, which did the genome of bats, 2 different bat species. Linfo Wang did that, and he found that they have a lot of they have a lot of duplication of genes involved in dealing with oxidative stress,
right?
Which happens when you fly. So they could be involved in the defense,
Yeah, and I wouldn't be surprised if Ebola viruses, especially since, as far as we can know, that if they are in bats, they're not killing them in huge numbers.
Right.
The biology of the virus would have to be very different in its reservoir host. So it could be aerosol transmitted, it could be sexually transmitted, it could be a variety of things,
or just contact, because they do live closely.
Or just contact. They're very social, they're clustered on cave walls in most cases.
Exactly, or in trees.
Yeah, so it probably behaves quite differently, if I may say. A virus behaves. Of
course, it's not alive, right?
Right, compared to how it is in humans.
Right, and I think of other viruses transmitted by bats too to humans, like Nipah virus, for instance. And that's a contact thing. You have to eat that in order to catch it.
Right.
It's on the surface of mangoes that pigs are rooting around eating, and all of a sudden a bat flies over and is scared off by another pig and defecates at the same time. And the next thing you know, you've got pigs to humans and all this kind of stuff.
Right. And looking at other viruses, I mean, flu viruses in birds, they're gastrointestinal, right?
Right.
Mm-hmm. They certainly are.
You've got a totally different route going on in the birds compared to what's going on in the humans.
Sure. Different biology.
Yeah.
So to address the other question, are there known cases of Ebola being sexually transmitted? I haven't been able to find anything in the literature, but the possibility exists. There's a 2007 paper from members of the Special Pathogens branch at CDC that describes the virus being found in semen, which we've talked about before. And in fact, one of the WHO recommendations is that for somebody recovering is recommended to avoid sexual intercourse for at least 7 weeks after recovery.
Do you have that paper in front of you?
I do.
Is it PCR or infectious virus?
Good point. We tested clinical specimens from laboratory-confirmed cases. Virus was detected by culture and or RT-PCR in 16 of 54 clinical specimens. I'd have to...
Culture, huh?
I'd have to look at which samples specifically had which, but yeah.
Remind me again, because I've forgotten, obviously. What is the incubation period? Do we actually know what that is?
Yeah, we do.
It's fast, I think.
No, it's not that fast. It is 14 to 21 days. I'm going to look at the CDC infographic from last week's episode to tell you precisely.
All right.
CDC infographic. Here it is. Dickson, can you see my screen?
I can. Yes, I can. All
right. Here's the incubation time. 2 to 21 days. Symptoms can appear after exposure.
Thank you.
And then when you get symptoms, that's when the virus starts to come out.
Right. So, I mean, I would imagine that shortly after being exposed to Ebola, let's say 2 or 3 days later, the rate of sexual activity during that very short window of time in an endemic area would not permit this to be a logical way of thinking about spreading it.
Yeah, I mean, you get very sick. Right?
Very fast, usually.
Things that are not compatible with
that's right.
sexual activity,
exactly
so probably not. But you never know, right?
You don't.
Go ahead.
I have the data now from Table 1 of this paper. The semen is virus culture positive.
Wow.
Oh, about that.
That's interesting.
For how long after
Now you're asking me even more details.
Yes, we're going to cover this paper.
Day after disease onset that the sample was collected, 40 to 45 days for one, 4 to 22 for another, so 7 to 45 for another.
Wow.
Yeah.
Okay.
Daniel Bausch is the first author. It includes John Towner, Stuart Nickel, Tom Kizek, Pierre Roland, and of the names I know, and others.
Over a month after onset of symptoms, if these people recover, you can still detect viable virus.
That's incredible.
All right.
That's very interesting.
Can you take the next one, Kathy?
Okay. Back to Jim, who writes, I wanted to comment on Dallas's letter. He's predicting the future and predicting doom. The Cambridge Working Group is also predicting a future of doom. Both sides seem to feel the other side is incapable of understanding what is really going on. So I want to make an aside here and say that he's referring to, I think, Dallas's letter that we read on TWiV 302. And to recap what I gleaned from what Dallas said, he was commenting at length about science policy issues, and he used nuclear power and accidents as an example. And in the end, Dallas cautioned about having non-expert risk managers sitting on grant review committees. So that's just to recap for those of you who might not remember Dallas's previous email. So back to what Jim's letter says. Nate Silver, in his book, The Signal and the Noise, Why So Many Predictions Fail But Some Don't, describes situations in which data has to be sifted to come to a conclusion on how to bet. If I understand him correctly, it helps to assign a probability to the prediction. This is a measure of how strong the data is. For example, would the Cambridge Working Group say there was a 10% chance that their predictions would come true, or would it be closer to 90%? Likewise, would those who favor Dallas's view of the system being overrun by bureaucrats with unreasonable control see this as 95% likely? As an outsider, I would give each side a 50% chance of being right. Would that the data were stronger. For now, each side has unsubstantiated anecdotal data and not much else, in my humble opinion. Both sides address this issue with great conviction. As I hear it, the data is puny on both sides. So far, I have not heard anything convincing on either side. Cheering for either side seems to miss the point that the data is poor. As Yogi Berra is reported to have said, predictions are difficult, especially about the future.
Indeed.
I love all things TWiV, TWIM, TWIP, and urban ag. I appreciate your tolerance for listener views which are different or wrong. And so I agree that we lack data, and in fact the Arturo Casadoval, Mike Imperiali, and Bio article specifically comments on the data that we lack, and they suggest certain things that can be done to get some of those data.
I would add that we have no known examples of lab-released pathogens wiping out humanity, and yet systems being overrun by bureaucrats is such a common phenomenon. There's actually something called Parkinson's Law, which describes why that happens virtually all the time in large organizations unless deliberate steps are taken to prevent it. Just saying.
I want to just also point out, I mean, that's fine. I agree with you in principle, Jim. However, scientistsforscience.org do not predict gloom and doom. All we are saying is let the experiments go forward.
Right.
And Cambridge Working Group, for example, does not want to do dangerous experiments, and without them, you wouldn't have candidate Ebola virus vaccines at the moment. That's why I don't think that's a reasonable approach. However, as Kathy pointed out, the MBio Casadoval Imperiali article did point out all the areas where we need more information to be able to make cost-benefit calculations, which we can't do right now.
Right.
So I think dogmatic statements that we should not be doing this are really not appropriate. Let's talk about what we need to do.
Yeah.
I want to point out also there's a new article by Casadoval, Howard, and Imperiali in MBio this week. It's called An Epistemological Perspective on the Value of Gain-of-Function Experiments Involving Pathogens with Pandemic Potential. And they basically say we should not a priori rule out gain-of-function experiments because they can have a benefit, and we have to consider that. So I'll put that link in the show notes. Maybe next time we can talk about it a little more.
Right.
All right. I'm going to do the next one, Dickson, and then you can do the last one.
Please
okay?
do. Please do.
Louis writes, be a little more, this is actually from TWiV.tv from last week's episode. Be a little more precise when you talk about transmissions. Ebola is not transmitted by the airborne route. That would be like TB and measles by particles suspended for prolonged periods in air. Aerosol is a bit less precise. When I cough, I can produce an aerosol of what I call the big chunks that can land in your eye or on your oral or upper airway mucosa and could cause infection. Airborne requires relatively sophisticated respirators like PAPRs, etc., and sophisticated isolation rooms with negative air pressure and specific flow patterns. See the CDC page on transmission-based precautions. The big chunks require contact and droplet precautions, i.e. face shields and surgical masks, or N95 masks at most, and infection is not by the airborne route. It is an important distinction. All right, so I disagree with this statement, all right? In the field of infection in virology, Fouché, Kawaoka, Palese all talk about airborne transmission by aerosols, because the aerosol is was defined last time. So Kathy gave us a link to a Medscape description of aerosol, and this is the following. Aerosol transmission has been defined as person-to-person transmission of pathogens through the air by means of inhalation of infectious particles. Particles up to 100 microns inside are considered inhalable or inspirable. These aerosolized particles are small enough to be inhaled into the oronasopharynx, with the smaller sizes penetrating deeper into the trachea and lung. Okay? And there's a little bit more there. But basically, an aerosol's droplet contains viruses, and they can be large or small, and that can determine, as we said earlier, how far they go. And that's how airborne transmission occurs. So it's all right? Am I right on this, or what? What do you think?
I think that he's maybe trying to make a distinction between airborne and aerosol, and I don't think that I do. I think of airborne being aerosol particles up to 100 microns in size.
Right.
And the big chunks that land in your eye are way bigger than 100 microns in size, and so to my mind those aren't aerosol, and it's still bodily fluid direct contact and not aerosol. So that's how I see it.
Yeah.
So I see airborne as distinct from direct contact, and somebody puking and some of the spray goes into your eye, I count that as direct contact, even though the spray flew through the air.
Yeah.
So another question I have then about the clinical picture of Ebola is that is there a symptom of coughing associated with the acute phase of this infection or not? It seems to me that what I've been reading is that
there's vomiting, Dickson.
There's vomiting, that's true. And that's a problem because that's huge amounts of fluid. But I don't think that there's much coughing.
So fever, headache, diarrhea, vomiting, stomach pain, bleeding, or bruising muscle pain. Those are the CDC symptoms.
Right, but if you think about the typical airborne infections and droplet-borne infections like influenza, there's a cough associated with that, which aids and abets the spread of that virus. So this doesn't have that modus operandus.
Yeah, but also, Dickson, when you cough or sneeze or talk, the virus has to be produced in certain places to be part of the aerosol droplets, etc.
Also true.
And also, if you're co-infected with TB, which is not being treated, because we're talking about this happening in some of the poorest countries in the world, then you would be coughing.
That's true.
Dickson, take the last one, please.
I will. I'd be happy to. Greg writes, hello. In listening to the letters about Ebola that were discussed in episode 302, it seems that more than one of the letter writers had concerns about airborne, in quotes, transmission of Ebola, yet used the non-equivalent term aerosol transmission. Although Kathy provided a very clear definition of aerosol transmission in terms of particle size and the requirement that they be inhaled, I think it might help your listeners to explicitly distinguish this form of transmission from more generic airborne transmission. Here's how I put it together. There are multiple potential routes for airborne transmission. One is aerosols. Another, as a letter writer mentioned, is coughing up blood. A third, technically, is throwing a beaker of infected blood at someone. That's pretty radical. The key difference in the epidemiology of spread, i.e. the R0, is the R0. As Vincent said, aerosol transmission can infect an entire subway car full of people after a sneeze. The non-aerosol airborne transmission that results from coughing blood or throwing a beaker would be expected to infect only people directly in the line of fire. Therefore, as far as we know, Ebola is transmitted between humans only by bodily fluids and tissue. This means that airborne transmission can occur in circumstances like coughing,
Body fluids.
he says fluid, but body fluids or tissue. But this is the exception. There is no known evidence that aerosol transmission has ever occurred between humans. I hope this helps. Best regards to all the TWiV team, Greg.
I'm sticking with airborne, meaning aerosol.
Yeah.
Yeah, I mean, this is how the field... I mean, I like
That's
your distinction, Greg. It's nice. But the
Yeah.
field has already used, aerosol.
And a particle size. They give it a definition for that, too.
Right. The field is already using aerosol and airborne interchangeably, and whether that's done in other fields, at this point, these are the terms that are in the literature.
Yeah.
Right.
I just wanted to put two links out there. Rich Condit in the Washington Post. This was the reporter that, written by the reporter that Dickson spoke with, Sarah Larimer.
Good.
And the article, Will the Ebola virus go airborne? And is that even the right question? And
Right.
I like Rich Condit here. He says, this is what bothered me more than anything else, and it comes from the title itself of the op-ed, is this idea that scientists are reluctant to discuss openly something or are considering it in private, said Condit. It fosters ... Condit added before stopping. Oh, this is so annoying. Yeah, so that was really good.
This is,
Right.
Yeah, quoted from that episode. Yeah.
And then I did an interview with Nell Greenfield Boyce of NPR, and she it was on NPR this week.
My wife called from the car to tell me, Vincent's on NPR.
Oh, cool. I was in Georgia. Monday night, I'm sitting in my hotel room. I get a phone call from Ian Lippin. He goes, Vinny, I just heard you on NPR. Thank you. It's just nice that people hear. All right, so...
And I just wanted to, this isn't the type of link we normally put in the show, but I just wanted to point out I read a really, really outstanding article just recently on someone's blog. It's called What We Are Not Afraid to Say About Ebola Virus.
Mm-hmm.
I think you've seen this one, Vincent, since you wrote it.
I did, right.
So if people are listening to this and still looking for more, this post on Vincent's virology blog, virology.ws, it really, I think, is extremely good.
Thank you. Yeah, it's funny. I posted it and went to bed last night, and this morning I got up and there were already 15 comments on it.
Yeah.
So it's
Nice.
stirring up emotions, I guess. Yes, Kathy.
I just wanted to quote the thing that I liked in the article from the Washington Post of Rich. It's the very end. It's not true, Condit said. It's not true that they're all worried. It's not true that they're keeping stuff under wraps. It's just not true.
Yes.
It does sound like him, right?
Yeah.
Yes.
That's right.
So that's it for our follow-up. And now we get to do a paper.
Hey.
Yes.
Yay.
We don't have to... yeah, it's good.
We should do an all-email episode.
Oh, boy. Yeah, email. That's what we have to do. All right, this paper is in PNAS. It is called Inhibition of Borna Disease Virus Replication by an Endogenous Borna Virus-like Element in the Ground Squirrel Genome. The authors are Fujino, Hori, Honda, Merriman, and Tomonaga from Kyoto University and the University of Wisconsin at Oshkosh.
Oh, wow.
Oh, gosh.
Oshkosh B'gosh. And this is all about Borna virus, which we have talked a little bit about before here on TWiV. Borna viruses are enveloped viruses with a single RNA genome of negative sense. And they infect a number of animals. The natural hosts are horses, sheep, cattle, rodents, birds. Whether or not they infect people is an issue of great debate. So there's some who believe that they do infect people and in fact cause psychiatric illness in people. And this was actually the virus that got Ian Lipkin started. Did you know that? Dickson, Despommier?
No clue.
So he actually cloned the genome because it was being implicated. Something was being implicated in these illnesses, and then he got his start as a virus hunter with Borna virus. And he continues to work on it today. They actually infect mice with Borna virus in his lab, and they do cartwheels when you infect them. It's really amazing. He's got movies of these things. Yeah. So I don't know what's going on, but anyway, the human involvement is weird, and it was first isolated in Borna, Germany, in 1885. Well, I don't know if the virus was, but that's where.
But we don't know if it was Borna there.
This is true. Well, it certainly wasn't Borna yesterday, that's for sure.
And a lot of horses died at that time, and this was the agent. So previously on TWiV, we talked about a couple of papers where, by analyzing the sequences of genomes of animals, like human and other animals, others were able to find little bits of virus sequences in them, not retroviruses, integrated into the DNA. And these include influenza, reoviruses, Ebola viruses, and Borna viruses. And the question is, what are they doing there?
Now, just before we move into the bulk of the paper, it's also kind of noteworthy about Borna that this is an RNA virus, but it can set up persistent infections.
That's correct. It doesn't
which is
kill
weird.
The cells, and it also replicates its genome in the nucleus.
Right.
Which is also weird. Flu is another one that does that, but it is weird, yeah.
For an RNA virus. I mean, obviously a lot of other viruses can pull that off, but...
Right. So what are these sequences doing there? Now, a couple of TWiVs ago, I was at the International Congress of Virology, and Carla Sally was there, and she talked about how in insects, when they get infected with RNA viruses, the RNA is copied into DNA by reverse transcriptase encoded in the insect genome, and then the DNA is used to produce short interfering RNAs to defend against infection. This is a really amazing story that she first presented at ASV this summer, and that's what is that, 301, right? So I asked her on the show, hey, there are other short pieces of viral DNA integrated in mammalian genomes. She said, well, I bet they have some role in defense, just like this. So, like a week later, I saw this paper. And
now, we've talked about the CRISPR-Cas system,
yeah,
right?
We have. I think so. Yeah.
Yes.
So, and that's a bacterial immune system, which works by a similar principle. It incorporates pieces of phage into a locus in the bacterial genome and then uses those. That's obviously been quite well characterized because it's now, you can buy kits to use it to delete your favorite gene. But, this is a phenomenon that at least bacteria were already using. So, it's a neat idea that metazoans can do it too.
Now, in the case of Borna, the fragments of DNA that are in the host are mainly from the sequence encoding the nucleoprotein of the virus, and that's a protein that coats the RNA genome. These negative-stranded RNAs are coated with protein in the virus particle. They also contain the RNA polymerase, because when the RNAs get into the cell, they have to be copied into mRNA in order to produce proteins. So this is mainly nucleoprotein. And the weird thing is that the open reading frames, at least part of the protein, are conserved. These things went into our genomes millions of years ago. And they're not only human, but you'll see in the paper, it's a squirrel, a 13-lined ground squirrel. Millions of years ago, yet the open reading frame is still there. Now, you wouldn't expect that, because if they don't have any function, these DNAs are just going to undergo mutation as the DNA replicates, and they're going to accumulate, stop codons and so forth. But these have an open reading, so that suggests they might be doing something. So there's some kind of selection to maintain the open reading frame. So in this paper, they study these integrated N sequences of Borna viruses from humans and also from the 13-lined ground squirrel.
Okay, here's where I get to pretend to be rich. Okay.
You're rich? You have a lot of money?
No.
Oh, I'm sorry. Yeah.
And I'll tell you all about 13-line ground squirrel.
Well, please.
So there are a lot of names for this animal, and that's typical linguistically for animals that have a broad geographic range. So they can also be known as striped gopher, leopard ground squirrel, squinny, and leopard spermophile. And that was in Audubon's day. And the 13-line ground squirrel is the namesake of the University of Minnesota Golden Gophers teams.
You're
Goldie Gopher.
kidding. So it
and
is
also,
Borna in the United States? Borna in the USA?
No, these are the squirrels. And it's responsible for the state of Minnesota's nickname as the Gopher State.
Wow.
But 13-line ground squirrel, also known as some kind of striped gopher.
So throughout this paper, they abbreviate this 13-lined ground squirrel as TLS, which is, as you're reading, it's easy to forget that, and then you're like, what is this TLS?
What's the TLS? When did they insert the TLS? Yeah.
So I always had to go back. But the real name is Ictidomus tridecimlineatus. Boy, they're hard to pronounce.
The real name is Goldie Gopher.
Anyway, these guys have an integrated piece of the Borna virus N protein coding region in their genome. How they figured that out? Well, I guess the genome had been sequenced and they found it that way. But it turns out that it's quite interesting, as you'll see. So they clone out these sequences into plasmids from the human genome and from the TLS genome, and then they put an HA tag on them so they can trace them. A piece of the hemagglutinin protein from influenza virus to which we have antibodies. So if you don't have antibodies against these N proteins, you can readily detect them in cells. And they put the plasmid in cells and say, where are the proteins in the cell? Now, normally N protein in a Borna virus infected cell would be in the nucleus because the virus is replicating there. So these things are made in the cytoplasm. They have a nuclear localization signal that gets them into the nucleus. Yeah. But these proteins, that the coding regions that they pulled out of the two hosts, go to the cytoplasm. Okay, if you just put the plasmids encoding either one in the cell, because they don't have an NLS, Dickson.
Okay,
right? That makes sense.
Yeah.
Now, if you produce, I have to really be careful to not say express the proteins. You'd have no idea how nervous I am.
I can see your hands. You're sitting there, it's shaking.
You can't express how nervous you are.
That's right. Ooh.
If you produce these proteins in cells infected with Borna virus, you find them in the nucleus.
Oh my God. OMG.
OMG. So that suggests that these TLS and human sequences, the proteins, are interacting with the Borna virus N protein, right? Because maybe they're being dragged into the nucleus.
No NLS?
Without a nuclear localization signal. Okay. But if you bind a protein that does have an NLS, of course you can get pulled into the nucleus. Sure. Are we okay so far, Dickson?
We're very okay.
Notice I ask you.
Well, because the audience is, I think, hysterical at this point, but that's okay. I don't mind being laughed at.
Why are they hysterical?
Because they know that I'm not a bona fide virologist. That's why. So I don't think like you guys.
Yeah, you do. It's all the same stuff. All right, so if you then ask, do these N proteins have an effect on viral replication? Okay, so the first experiment they have cells persistently infected with Borna viruses. In other words, you can put virus in cells and they get infected and they replicate, they divide, they don't die, and they keep producing virus. And if you introduce these plasmids, including the human or the TLS N protein, what does it do? And they measure viral RNA genomes and they measure viral mRNAs, and it's depressed a bit.
So I just stop for a moment to clarify something for me. When there's a persistent viral infection, this places a metabolic stress on the cell in question. Yes, because now it has to produce not only itself, but also something in addition to itself.
I presume so. Yeah.
So does the metabolic rate of the cell increase, or does its division rate decrease?
Those are two different questions.
No, they're the same.
Does the metabolic rate go up, or does the division
If it goes
decrease?
up, then the division will stay the same, because it can accommodate for both itself and its parasites. Or does the actual rate of division slow down because it's compromised?
I don't know. Don't know the answer. It's a good question, Dickson. Okay, so you do think correctly.
But I just thought I would ask that as an energy balance type person.
Yeah,
I was just interested in the ecology of this thing.
I would guess now that the cells continue to divide, but I could be wrong, right?
Is that true for persistent infections?
Well, some do and some don't. Yeah. Right? It really depends. Now, looking at the results here, you're not impressed because you go from 100% to 70% of the amount of RNA, either genomes or mRNAs. So that's not huge. So I looked at this initially. I said, meh.
Nah.
Nothing but...
It's a New York term.
Meh.
That's right.
Meh, right?
That's right.
Is that a New York term?
Yeah, it is.
Do you say that in Michigan, Kathy?
I've heard it here.
Yeah.
Now, however, if you do a different experiment, now you infect cells with virus, and then they have cells stably producing these TLS and human N proteins, right? and then they infect those with Borna virus. And you get a substantial inhibition of virus production. They look by immunofluorescence. They have a green fluorescent labeled virus that they can use. And if you look at the micrographs, and there's really, it goes from a couple of foci of infection to one or none when they put these different N protein in these cells. So it's quite impressive. What I found neat is that the inhibition is also achieved with the N protein of the virus itself, because they do a control where they take the N protein, clone it, and produce it in cells, and that also inhibits as well. That's in the figure. Not as well as the others, though. It's quite interesting. So there's something special about those. You can also take an infected cell, a persistently infected cell, and mix it with these stable guys that are producing the N protein, and they don't get infected. So these are protecting these N proteins from the squirrel. I'm sorry, the human one doesn't work. I should have mentioned that. The human N protein, cloned out of the human genome, doesn't protect against infection. The ground squirrel one does. And so this is a puzzle which will remain unsolved throughout the end of this. Right?
Perhaps.
Now, the N protein is part of the polymerase complex, so they have a nice polymerase assay where they have a small genome with a reporter, and they can put this in cells and assay viral RNA replication, basically. And the ground squirrel N protein inhibits that. And that it seems to be making a complex with the viral RNA polymerase, because if you immunoprecipitate with an antibody to the ground squirrel N protein, it brings down the complex, and you can see there's viral RNA in it, and there's the P protein, which is part of the polymerase complex. So,
one other question, in terms of an ecological situation at least, and this is obviously a wild animal that this was isolated from. How many other groups of animals would you find Borna virus infections in?
Yeah, there are quite a few. There are horses, sheep, cattle, rodents, which is a lot, right?
Okay.
There's a lot of rodents. Birds as well.
You bet.
A lot of different animals.
Okay.
So, yeah, you could ask, is this present similarly in all of those?
Yeah, and I mean, maybe we lost our Borna virus infection because this protein works so well.
So our human DNA doesn't inhibit infection. Only the ground squirrel one does.
But it might have inhibited our version of the Borna virus at some other time.
Well, I don't know what our version would be, Dickson.
Exactly.
That's the thing.
Well, that's the point.
It's controversial whether people are being... I'm not sure it's ever been isolated from a human.
Or it inhibited the Borna viruses of an extinct ancestor.
Exactly.
Maybe.
Exactly. That's
because these are conserved, but how long would it would take quite a while to to start accumulating mutations in a long conserved gene if it was useful just 50 million years ago.
Yeah. Now, so the idea is that this N protein is binding to the so the N proteins on the viral RNA, the viral N proteins, they interact with each other. They form multimers, and so the idea is that this is participating in that, but it's missing sequences that will allow it to be functional. So it's inhibiting. It's a dominant negative inhibitor, basically. Yep. And again, it works only the protein from the squirrel, but not humans. And they suspect that maybe so. But the weird thing is that the open reading frame in people is still conserved. So why is that? And as Alan said, maybe it hasn't been long enough yet to mutate. But they suggest that it may have some other function. And
does the Neanderthal genome have this?
Good question.
Yes, very good question. Can you look, Dickson?
I will promise to do that someday. As soon as I clone my own genome.
We're like 3% Neanderthal, right?
Oh, well, some of us are more than that.
Dickson,
no, serious.
Okay, here's the answer. The human EBLNs, these Borna L DNAs were generated 40 to 45 million years ago, which is before Homo sapiens, obviously.
Yes.
It's way into our precursors, right?
Correct.
So probably.
Yeah, that's right.
When did we diverge from rodents?
I never did.
I was awfully young at the time, so I don't remember.
Some of us have remained rats.
I think that's. I don't know.
Well, rodents were 65 million years ago. They were the survivors. from that Holocaust from the meteor. So supposedly only the ground burrowing mammals were the groups that actually survived that group of Earth events, which extincted most of the land animals at that point.
Right. So we may just not be far enough from rodents to have accumulated a lot of mutations in this.
That's a good point.
Seems like it's far enough.
60 million years is a long
years
time.
should be enough. Now they say here that there's evidence that this gene, this N gene, is actually a tumor suppressor for cervical cancer in humans.
Oh,
goodness.
Well,
There's
that's another
evidence.
possibility.
Goodness.
If it picked up a new function, then it would certainly be conserved.
Right. So that's pretty neat. It's got a new function.
We don't throw anything away, do we?
Well, retrovirus genes have acquired functions in us.
Sure.
I'll be darned.
So why not? So this is pretty neat.
Evolution is bureaucratic.
Tell you.
This is interesting. So this is at the protein level. When I saw the title of this, I thought, oh, these are making interfering RNAs, but they're not.
They're not.
So maybe some of the others are. So I mentioned earlier, we have pieces of Ebola viruses in our genome.
Right.
What are they doing there? Well, they're not protecting us, I guess,
No.
right?
No.
Who knows what they're doing?
Are there any therapies that are planned based on this strategy for other viral infections, perhaps?
What strategy is that? Producing a negative dominant protein?
Exactly.
Oh, absolutely. Oh, dominant protein.
Protein
I'm sorry.
is tough.
Yeah.
Therapeutic proteins
negative
are hard.
RNA, definitely.
But RNA for sure.
RNA, sure.
For sure.
Okay.
I have the factoid about divergence time between human and mice about 96 million years ago.
It's a while back.
Yeah, it's long enough to get changes.
while
Something's
back.
going on here.
Maybe we acquired it from them recently via a lateral transfer.
That's another possibility. Maybe we got their Borna viruses.
Exactly.
Hey, we have time for a few emails.
Uh-huh.
We can make inroads into this.
Yes.
First one's from Basel, who writes, Dear mutual vaccination proponents, I'd like to share with you a story that happened to me this past weekend that I thought is worth sharing. As a first-time expecting parents, my wife and I took a birthing class among many other parenting naive parents-to-be. Parenthetically, we took those, and in the heat of birth, it all goes out the window.
This is all true.
My wife is like making, she's in pain, and the guy says, "Didn't you take your birthing class?" And she said, "I feel like punching you."
Your birthing class. Come over here. I'm going to show you what I learned.
The birthing instructors gave a fairly balanced discussion on the pros and cons of many topics, such as epidural versus non-medicated birth, vaginal versus cesarean delivery, etc. Even what weirdly to do with the placenta after birth. Take it home, dry it, and drink it.
Oof.
Now, if you're Carolyn Coyne, you do experiments with it. What ticked me off the most was that when it came to vaccinating your newborn, they stepped back claiming that it's a parenting decision we need to discuss at home. I thought the least they could have done is provide and discuss the pros and cons of vaccination, if there are any cons for that matter.
Yeah, maybe that's why they didn't do it, because they couldn't think of any cons.
Or they're anti-vax and somebody told them not to express that.
I didn't like that. All attendees were new expectant parents, and the main reason they were in this class is to learn. And what's more important than discussing this topic, especially in Portland, Oregon, our fair city, as click and clack, the Tappet brothers say, with the surge in measles cases. I thought if there is any class to discuss this topic, it has to be birthing classes and newborn care classes. We're taking a newborn care class in a couple of weeks in July, and I hope I don't encounter such an unpleasant scenario. Yeah, that's really... I would have said, "Hey, why can't we discuss the pros of vaccination?"
Yeah,
of course.
Yeah. Hopefully, Basel felt empowered to do that in the newborn care class. If it wasn't brought up, that he at least brought it up.
Yeah.
Kathy, can you take the next one?
Sure. Christoph writes, Dear TWiVers, I'm a long-time listener and have patiently been waiting to contribute my 5 cents, so here it is. In TWiV 289, Vincent stated that he does not believe anything eats viruses. I have to disagree. Check out this contribution from Juan Gonzalez and my PI, Curtis Suttle. Gonzalez, and he gives the link. I guess it's Gonzalez and Suttle is the paper. Turns out viruses make up a small but significant proportion of a balanced phagotropic nanoflagellate diet. Also bear in mind that this study was pre-megavirales. I'm sure a mimivirus or a large Ficodna. Yeah, there's too many vowels in there. Okay. Makes a welcome meal for those little flagellates, and they are abundant. And he gives a paper for that entitled Exploring Nucleocytoplasmic Large DNA Viruses in Terra Ocean's Microbial Metagenomes.
Interesting.
And he gives a link for that. Best, Chris. P.S. The weather in Vancouver, BC is currently 18 degrees C, 64 Fahrenheit,
Nice.
sunny with humidity of 24% and no wind.
That's what it is here, Dickson.
That's lovely.
Yeah.
It's funny. I asked Curtis, because I had him as a guest at the International Congress of Virology a couple of weeks ago, and he mentioned his work. And then we had this letter. I thought that was pretty cool. So
Nice.
it's on TWiV 301.
Very nice.
Dickson, you want to take the next one?
I'd love to. Richard writes, Dear TWiVers, I refer to TWiV 281, The Salk Legacy with Peter L. Salk, and I would like to offer a somewhat different interpretation of Jonas Salk's famous comment about patents and the sun. In 2002, I was asked by the Rockefeller Foundation to examine the extent to which patents, intellectual property, IP, limit access to important health technologies for the poor in developing countries. Our study led to the conclusion that achieving access to these technologies requires attention to research and development, establishing high-quality manufacturing, preparation of domestic and international markets, management of intellectual property, and establishment and implementation of high-quality regulatory systems to ensure safety and efficacy. Through a number of detailed case studies, we also concluded that while intellectual property management is one of these requirements, it is usually not the most important and is often of little importance. However, when it is important, it is very important. Further, the analysis led to a counterintuitive conclusion that intellectual property is really important when regulatory requirements raise the cost of development to very high levels, for instance, $100 million. Under those conditions, no company will invest the required funds without intellectual property protection. Thus, the granting of intellectual property ownership is one means to help raise the funds necessary to ensure safe and effective products. This analysis led us to have a somewhat different perspective about the patenting the sun comment in the light of the subsequent Cutter incident. TWiV is proving to be an extraordinary means for airing important topics that affect a spectrum of the population, from parents deciding whether or not to immunize their children to companies deciding whether or not to develop a vaccine. You're doing a wonderful job. All the best, Rich.
So what was it? They asked him why you didn't patent his vaccine. He said, would you patent the sun?
Right.
Well, that was a different era, right?
Well, yeah, and regulatory requirements were lower, and the initial investment to develop the vaccine was not was not out of reach for the company, even without patenting it. But regulatory requirements were lower, and we had the Cutter incident, and now regulatory requirements are higher.
Alan, I saved this one for you.
Oh, good. This is an interesting letter. It's the way it's formatted.
Sounds like a Gilbert and Sullivan play.
It's like a poem, and no capitalization.
That's right. E. Cummings is writing.
So it's E. Cummings is Kathy's note here. So John writes, "I'm scared of science. Trust me, I'm a scientist. I'm scared of being treated like cattle. Scientists use the term herd immunity. I'm a scientist, but don't trust myself, so we'll have a blind experiment. Better make it double blind. That is how much I don't trust myself, and wait for my peers to replicate it. But trust me, I'm a scientist. I'm a scientist working for the arms industry. I'm a scientist lobbying for government patronage. I'm a scientist with a lucrative patent. People who are not trained to think scientifically are still problem solvers and very intelligent, so they will solve their problems without scientific training. My child is healthy. Why invade them with a vaccine and subject them to a needle? Trust me, I'm a doctor."
Question mark.
Trust me, I'm a doctor.
Or trust me, I'm a doctor.
You cannot fake science, but you can fake scientific arguments. Lay people know this. Scientific training is a matter of social policy and education systems that 90% of the population can be intelligent and productive members of society with highly effective problem-solving skills, yet unable to evaluate the specialized language of science and then yet the more specialized language of medicine. You cannot have specialism without the trust of those who are not specialized. You cannot hide behind science as being pure and altruistic when it's sponsored by a vested interest. You cannot raise your hand and say, "Trust me, I am the good scientist and not the bad one." You cannot bridge the gap of science versus not science by media and communication skills. You either are scientific or are not. You either have a population educated in science or you opt for specialism. I love your podcasts for being better than the rest in objectivity and prolonged debate, with both humor and humility, diligence and controversy. Own your own frustration at being specialized. Keep communicating. But do not express frustration at people who are scared of you. They have the same brain size as you, and you have the same emotions as the people who are scared of your specialized knowledge. Mistrust of science is not a failing. He's capitalizing now. Mistrust of science is not a failing of those who mistrust and not a sign of ignorance, just normal people without scientific training needing to engage with information they are not educated to process. Problem-solving behavior is a problem in itself and not necessarily a virtue, yet scientists, environmentalists, and health workers are often the last to know that their need to solve the problem is a need, an emotion. Hysterical people find it hard to hide their behavior, but obsessive people can hide their emotional need of safety through problem-solving by specialism, procedure, productivity, rationalism, etc. Is there such a thing as too much objectivity? Is there such a thing as too much biodiversity? Is there such a thing as too much intelligence? Is there something that is not the environment? Is there something that is not nature? My point is that science, environmentalism, and medicine are clothing themselves with belief structures. Environmentalists versus skeptics, doctors versus the anti-vaccine lobby, Darwinism versus the religious right wing. Giving the climate a control mechanism, yet expressing the need for nature to be free and wild. You are an ivory tower as scientists. You will feel loneliness and isolation in your knowledge as scientists. You will have exaggerated emotional perceptions of control and solution. You will have incredible self-imposed expectations to solve problems and express frustration towards those who don't share your insight. Please keep trying to communicate. You're doing a fantastic job. Don't get frustrated at people who are scared. It doesn't work. Most people have the same brain size and are also trying to problem solve, but own the self-interest and betrayal of your colleagues when science has led us wrong and into danger. Trust is a two-way process where science is proved untrustworthy. Scientifically, humans at one point thought the most efficient way to dispose of each other was by gas ovens and use their waste products to make pills and soap. Intelligence is what you make it through emotion and is very dangerous without. Once again, please don't feel discouraged despite my gloomy email. Thanks again for your often startling and informative efforts. My problem-solving need was to respond to your stimulating podcasts and receive some feedback to validate my intellectual process, but I'm feeling guilty of writing such a long email to join up and solve everyone's problems. Best regards, John. Very stream of consciousness, but I think I get where you're going with most of that.
Yeah.
So the one thing that I noticed here, when our colleagues make mistakes, we have to own up to it, right? And not defend them.
That's right.
And I think we do that, right? We certainly
I think we're pretty good about that.
Pointed out the MRV issues right away.
Of course.
Right? Our skepticism with it.
But only the people who didn't fake the data. We were not too kind to the people who did.
No, but when that was found out, yes,
yeah.
But we didn't know that from the start.
No, that's right. But you can be very harsh on those people because they give everybody else a bad reputation as a result.
Yeah.
Right, and I think we're pretty critical of papers we present in general. I mean, today's was fairly straightforward, but in many cases we'll point out issues with particular studies, even if we like the overall work. And I think that's actually an important part of the show. I would just refer to Vincent's repeated statement, which is trust science, not scientists.
Well, I think I was the one that actually came up with that one.
Was it?
a long time ago.
Sorry.
No, that's quite right. That's quite right.
Okay.
No, but the other thing is that science is a work in progress. It's never finished. It never concludes ultimately that this is the end of anything. And I think that's the reason, Alan, that you gave us as to why you love science so much.
Exactly. Keeps going.
And it keeps going and going. And so as much of a confidence level that you have in some data that says this is the way it is, it's always possible that at the next time you try this in another way, it's going to turn out different.
Right.
And the other part here is that people are scared and we shouldn't make fun of them or anything. But, the thing is that most people are scared or maybe they don't know. There's a very small minority that's vocal and says dogmatic things. Sometimes it makes you think that's the way everyone is out there who doesn't understand science, but it's not the case, right?
I can think back over the Monty Python episodes that I've listened to, which expresses that very, very clearly in terms of medieval thought and adopting certain hypotheses that are today seem ridiculous. But in those days, they were actually taken very seriously. And then to hear it expressed in a humorous way is it'll be amusing if we could jump into the future and see how we're perceived in 200 years from now. Maybe they'll do a Monty Python about us.
If you're lucky, Dickson.
Well, I mean us in general, the scientific community in general.
I think I like to think we have a little more insight than they.
Yeah, me too. Yeah,
at least we're not saying we know all the answers.
Right.
So that's some progress.
Oh yeah, that's absolutely true. But there are some scientists that are hooked on their own hypotheses and refuse to give up no matter what, and that's crazy thinking, I think.
I like your email, John. Thank you.
Yes.
I think it's well done. And I think we'll just do one more here from Nicole. Dear TWiV crew, in case you haven't seen this, she sent a link to an article in, it's called Anti-Vaccine Movement Slammed by Daily Show. ID doctors, pediatricians, happy.
Right.
And this is just coming up here. This is in the New England Journal Watch. All right, so it's an article there. In HIV and ID observations, Dr. Paul Sachs shares an anti-vaccine movement video that makes him very happy. And Nicole writes, the anti-vaccine crowd gets a pretty good drubbing from The Daily Show. Dr. Paul Sachs would feel bad for them if he weren't an infectious diseases specialist married to a pediatrician. See more in his HIV and ID observations blog. Yeah, so he's got a blog of his own, which is separate from this article. So that's pretty cool. Check that out. It's sponsored by New England Journal. And
The Daily Show is not sponsored by the New England Journal.
No, the
blog is.
The blog is sponsored by the New England Journal, or is part of it. Yeah. And Nicole finishes, I can't say enough good things about your podcast. Thank
you could try.
I was going to say that. Alan, you took the words out of my mouth. Give it a try, please.
We had a lot of good praise today. I appreciate it. Yeah. I'm glad people like us. No, we don't get everything right, and
no, certainly not.
May not agree with what we feel about things, but that's fine. Science is all about discourse and discussion, not dogmatic stances, right, Dickson?
That's very true.
Let's do some picks of the week. I bet you don't have one, Dickson.
I
right?
do. I picked it last week, but we didn't give a link to it. But we can this week. This is the robot link.
The robot link.
Yeah, it was in last week's science.
Yeah, you said you were going to send me the link, and you didn't.
Well, I can do that right now because I've got the damn issue.
I felt so badly, Dickson. You didn't.
I have the issue on my desk outside. I'll give it to you.
We've got the link. It's already in the show.
Yeah,
it's already up at the top.
There. It's there.
Kathy put it in. She did your work for you.
Thank you, Kathy. No, she didn't do my work for me. This is this week. That's not last week.
She found the link. Found the link. I'm sorry. You lost it.
I'm getting very defensive here. I shouldn't do that.
Because I pick on you too much, right?
No, I'm the pick of your week.
Hey, that's very good. My pick of the week is picking on Dickson. Excellent. Kathy, what do you have for us?
I picked a blog post about whether or not you should refrigerate your tomatoes, since this is pretty much the peak time of summer vine-ripened tomatoes. We got this with our CSA share this week. Our department has a community shared agriculture thing that Alice Telesnitsky started, and we get flowers and fruit and stuff and recipes and not fruit so much as vegetables, but unusual things. And she sent this link where a chef has done a study to try and figure out, should you or should you not refrigerate tomatoes? Because the dogma is that you should not. But then he actually says that most of that was done on sort of hothouse and commercially grown tomatoes, and he does some tests. And it also depends on what your room temperature is.
Right.
And so it's a really interesting article. And the bottom line is, if the temperature is moderate, like we all have outside today, then having them on your counter for a day or two to continue to ripen is good. But then putting in the refrigerator is fine. And then let them warm up to room temperature before you eat them for the best flavor.
And actually better than fine. Because if your room temperature is, if it's summertime and your room temperature is like 78, then they rot very fast, they found, and he found that putting them in the refrigerator, they stay much fresher, much longer.
This is true. And it depends on the kind of tomato, too, I think. The grape tomatoes.
No, actually, this blog post...
Really? All tomatoes?
Well, the ones that he sampled, which is, I gather, a wide selection. He's got a shot of a bunch of different tomato varieties in a box, and talks about collecting all these different freshly harvested varieties. And
he talks about who has sampled them and the numbers of the results.
It is nicely done. This should have been in a peer-reviewed journal.
Now, I grow my own tomatoes, and sometimes they fall off before they're red.
Yeah,
so I bring the green ones inside, and I leave them out for a week or 2-3 weeks, and they get red.
How about that?
So I guess if I put those in the fridge, they wouldn't get red.
I don't think so.
Right.
You know, this is what I miss here at Columbia, Kathy. You guys, you have this community. You all live around the same area. You do stuff like that. Here at Columbia, everybody goes miles and miles apart. I never see any faculty anytime except during the day. And I really miss a faculty community.
I agree.
You know, so if I were to do my career again, and this is advice to any of you out there who want to be in academia, I would go somewhere where there's a community. You know, like a Michigan or a Princeton or a Cornell, where people are all, you know, in the same town.
Notre Dame was like that, too. I was a graduate student here as a master's student, and then went out to Notre Dame for my PhD, and what a vast difference in sociological behavior. I mean, it was just amazing. We had a party every weekend. We had gatherings of faculty and students and the graduate students, too. And it was just very, very social, and that was totally...
Well, I actually found at the graduate student level... Columbia was a very social place in the postdoc level.
Yeah, you guys are all living around here.
Right,
yeah, because all the grad students and postdocs either live right around the Columbia Medical Campus there in Washington Heights, or around the downtown campus at 116th. And so, you know, and we had every Friday we would have a little beer, beer served in one of the lecture rooms. Everybody would get together and.
Okay.
You know, very, very, very social group there. So an urban university can be fine for that if everybody's tends to cluster right around it.
And Rockefeller was like that, too, when I was a postdoc.
Rockefeller, of course, and they actually, unlike Columbia, they actually subsidize their student housing, so you can afford to live there.
Yeah, that's right. That's right.
And they have also big faculty buildings there where they all live. So you can see other faculty.
And they have a faculty club, which is very accessible.
But here, you know, Columbia doesn't really help us with our housing.
On the downtown campus, though, there is faculty housing. There is gatherings there. So
it's hard for us to live down there, though.
We're isolated. On our salaries?
No, our medical school is not, you know, we're not allowed to get pick of those apartments. It's very difficult unless you're threatening to go somewhere else. It's hard to get one of those apartments downtown. So I would say, downtown faculty may have more of a community than we do. I really miss that. And and that, when you said that, Kathy, that kind of reminded
Vincent
me.
Turned green. I just noticed he was turning green.
No, I'm okay.
But if we leave him out on the shelves for a week, you're just going to ripen.
I'm okay, but just saying, for people who are thinking about it, it's an important thing. Make sure there's a community, you know.
You bet.
Alan, what do you have for us?
I have another photography pick. I know I do these periodically, but this one is it's a cool organization of an archive that was already online, but the way they've put this together is really, really nice. It's called Photogrammer, and this is based at Yale. They took these photographs from the Farm Security Administration and the Office of War Information. These were programs in the 1930s to early 40s when the U.S. government employed a bunch of photographers and writers to go around the country and document the way Americans do things. And that was obviously a large part of that was trying to pull the nation out of a recession or depression. But the upshot of it is some really, really great documentary photography from all over the country from a particular slice of a very transformational slice of time. And what these folks have done is they've organized, there's something like 170,000 images in this database. So if you go to Library of Congress, you can look up these images, but where do you start? You know, it's just, it's so overwhelming. So this thing has a map that you can click and a timeline, and you can move through the timeline and select which portion of time you'd like to find. And then you can select a point on the map, and it's got the it highlights on the map where they have coverage. So I'm just going to go to southeastern Michigan here. So I've got Wayne, Michigan. They've got a couple hundred pictures. Washtenaw, Michigan.
That's by county, because Washtenaw is our county.
Washtenaw is your county. Okay, Washtenaw. So we've got 16 pictures from Washtenaw. In
a certain time zone.
In a certain time period, and I've got them in 1941, and it's just people doing various things. This, of course, since it's 41, it's a lot of defense industry.
Sure.
Documentation and
amazing.
Is it cross-indexed with the photographers also?
Yes.
Got
You can
their
find
names, yeah.
you can find who the photographer is for each of these.
And you can select one particular photographer and see his
Oh, wow.
or her
Yes.
photographs for that time.
Outstanding.
And place.
Yeah, that's cool. I
Outstanding.
just found Union County, which is where I live,
Right.
and there's cool photos of people working in the General Motors plant in Linden, which is no longer there.
Yep.
Very cool.
And these are all public domain images, by the way, because they were produced by government workers.
Look at
I'll be
this,
darned.
a welder trainee. Wow, this is great.
Yeah.
All this stuff we don't do in this country anymore.
Yeah.
That's
Well,
very
now you can find.
We
lots
have
of
everybody
photo
putting it on Flickr
Everybody's
and
putting
Instagram.
everything.
Yeah,
Photo
of course.
documenting everything. It's
Yeah.
not
true.
a problem. But
You hear it, Dickson? You better document yourself.
Yes, sir.
My pick comes from my Australian visit. I went to interview Peter Doherty, who is an immunologist and a Nobel laureate. I went to his building. It's a brand new research building called the Peter Doherty Research Institute.
Oh, my God.
Lovely. He has a lovely office with a wall full
Can
of
imagine.
memorabilia. Yeah, he's a great guy, though.
Yeah, he is.
Anyway, I had a great talk with him. So I did 26 interviews for our new textbook with different people for each chapter, and he was one of them.
wow.
And I'm hoping someday I might be able to release them on TWiV because they're pretty cool. But he had a pile of books on the desk in front of us all that he'd written. He wrote them all.
A pile of
And he
books.
gave me a couple. And one of them that he gave me, I read on the way back, is called The Beginner's Guide to Winning the Nobel Prize, Advice for Young Scientists. And it's pretty good. It's a lot of fun. It's all about his life, how he got into research, what he did, how it works, and he talks about some of the other Nobel laureates that he knows and so forth. So it's pretty, he wants to write. He has been doing this for a while, and he wants to continue because he's really not doing research anymore. He wants to write so that people understand the science in an easy way. So it's written quite simply.
Isn't that why you podcast?
Yeah, sure.
Okay.
I mean, there are many ways that you can do this, right? And everybody, a lot of people should do it, not just one. So we need to have a
Indeed.
full court press to the public to see all this science, right?
Look at that.
Anyway, he's a very good writer, and he's a lot of fun. He's got a good sense of humor, so you might want to check that out. If you're thinking about winning the Nobel Prize, get his advice. Why not? We have a couple of listener picks. One is from D, who writes, "Hello, TWiVimps. Virology, microbiology, parasitism. I listen to all three. Long-time listener, but I never thought I'd ever write, as I am not in your fields. My science education is limited to high school biology, and I generally don't understand anything when you all get super technical, but I pick up on general themes and really enjoy the conversational learning. Having listened to you guys for this long, I often think I would be a good lab geek. I currently help folks with their technology, mainly a computer geek, and in my past life worked in the music business getting records on the radio, known as record promotion. In addition to the many tech podcasts I listen to daily, I also listen to a variety of non-tech ones as well, including yours. I was turned on to TWiV from Futures in Biotech, then TWIP and TWIM, although I am a year or two behind on all three. I was disappointed I missed you in Denver last year because I'm so far behind."
Well, we're about that far behind on our emails. So
And also, what you could do is just do what I did when I started listening to TWiV, is listen to the current ones and then fill in with the back ones. And then you never know whether some topic is 2 years old or very, very current, but it still works.
That's a good idea.
Also, we could listen at 1.5 or 2x. Some people do
Right.
that, right?
yes,
One podcast I listen to is This Week in Startups, known as Twist, with Jason Calacanis, which discusses tech startups and helps young entrepreneurs with theirs. On a couple of episodes, I've heard your frustration of papers published behind paywalls. On the February 11th, 2014 of Twist, Richard Price of academia.edu talks about this issue, and I thought it might be of interest to you. So academia.edu is a place where you can put your papers. It's an interesting model. You probably know about this already, but I thought I'd do so as well just in case. Thank you for all your time and efforts in putting all three podcasts together. So I know Jason Calacanis because he's on some of the tech podcasts that I listen to, and he started this podcast, This Week in Startups, and his goal was to make $1 million a year with it. And he made $500,000, so he canned it. He said, I didn't reach my goal. But he was making $500,000. He had a whole bunch of different podcasts. It was kind of a podcast network, so really interesting.
It sounds like a self-fulfilling prophecy, though. If it's not a startup, it ends.
Well, he talks about business. And our other pick is from Basil, who we heard earlier. He writes, my pick of the week is a fully functional microscope built from Lego parts. This microscope concept is submitted to LEGO, and upon receiving enough votes, LEGO will manufacture the kit and sell it in stores so that kids and adults alike can build their own microscope. So I'm hoping that this pick gets the TWiV pump and people vote for it. So he provides a link to the scope and also where you can vote. So TWiV Army, go out there and vote this up because this is a cool thing.
This is neat.
Yep.
Kathy likes it.
It really combines two TWiV memes, the Lego figures and microscopes that you can build. So I thought that was really great. And I went to the site, and you can open an account so that you can vote for it. And then you can click that you want to track the progress of the particular thing that you're voting for. There's also one that I saw, a biology lab. And it shows double helix of DNA and some other things. And be prepared to answer questions about how hard you think it would be to assemble that particular thing. Because once you start doing the survey, you can't see the thing anymore. But anyway, it's a cool thing. So.
All right, so thanks for that. And that will do it for TWiV 303. We are marching towards 400, Dickson.
We are.
And I just want to remind everyone of the sponsor for this week's episode, the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. Great place to do research if you're a graduate student thinking about getting a PhD. They have a December 1st deadline. You could take a look at their department at mssm.edu/MIC. We thank them for sponsoring this episode. And as I said before, if you'd love to sponsor a episode of TWiV, it's not very expensive. You have a department somewhere or a product that's relevant, and you'd like to grab our many, many listeners.
You want a bump?
Yes, get a TWiV bump for your product. Send an email to me at twiv@twiv.tv. You can find this episode and all the others at twiv.tv, also on iTunes. And we love getting your questions and comments. One day we will get to it. We promise. Send them to twiv@twiv.tv. Dickson Despommier can be found at verticalfarm.com. Thank you, Dickson.
My pleasure. I really enjoy this.
Do you?
I do.
I'm sure you do.
No, don't I sound like I enjoy this?
I'm not being silly. I know you like it.
No, I love it.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thank you. This was a lot of fun.
Alan Dove can be found at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.